Hello and welcome. So we discuss about the STS page gel system for Western blotting, and we talk about the vertical gel electrophoresis system. So in vertical gel electrophoresis system, there are two type of gels that are casted, the stacking gel and the resolving gel for the optimal resolution of the proteins that have been loaded in the wells. So stacking gel has a different concentration, for example 7% concentration of bisacrylamide and acrylamide, a different pH, it has got a lower pH, a larger pore size and different ionic content. The system is also a discontinuous buffer system where the primary anion in the gel is different or discontinuous from the primary anion in the running buffer. So this discontinuous buffer, for example, one of the system is the lamellae system which contains the tris glycine is an example. So as you can see, when we load the samples in the gel, then the different anions sandwich the protein complex in between. So this is the three kinds of ions which are used. So the tris glycine is the buffer system which we are using. For making the gel, we are using tris chloride. So tris is a common ion in both the gel as well as in the buffer system, but in the gel we are using tris chloride and in the buffer system we are using tris glycine. So the chloride runs in front, in between runs the tris protein complex, and then the trailing ion is the glycine ion. So this creates a tight packing or stacking in the gel and then better resolution as it enters into the resolving gel. So this kind of a system is termed as the discontinuous buffer system, and because these different ions are encompasses or covers this polypeptide chain during the stacking period, and then therefore the resolution is resolution is better when it comes to the resolving gel. So as I said, the gel buffer system, three different ions are basically used. One is the chloride ion, which is the leading one. It is supplied by the gel and it is the leading ion. Then there is the second ion which is called as the tris ion, which is basically associated, or this is the glycine ion which is the trailing ion which comes later on and it basically trails the protein complex, and then the tris is the common ion both in the running gel as well as in the gel buffer. So this is termed as the discontinuous buffer system and an example of tris glycine system for the optimal resolution of the proteins that we have loaded. This is a photograph shown here for the output of the STS page gel. So as you can see, different protein bands are shown here. So this has been stained with Kumasi blue and we can observe the different proteins. This is a total protein lysate that has been run through. So where proteins of different sizes have migrated along the gel in response to the applied voltage, a size standard lane is on the right side of the gel. So a size standard is also run. So this is the typical output of a SDS page gel. In this we stain it and we de-stain it also, because first of all the stain takes up the entire gel and then we de-stain it so that only the protein bands are remain stained. So the de-staining is an important step here. So for the sample preparation to electrophoresis, this is the entire series. You start with the lysate, you isolate the protein from any cell or tissue or whatever conditions you have done. You isolate the total lysate, you set up the gel, you load it on the gel, you run it in the voltage, and then this is the output that you basically get. The next aspect is the electroblotting. So this is the blotting part. Now we are ready with our gel. Now we need to blot the proteins onto the nylon membrane. So just like we did for the southern blot hybridization, here also we do the blotting, but it is not done manually or by the capillary action. It is done by the electroblotting. As I said, electroblotting method is very speed, very reliable, and it transfers most of the proteins that are present on the gel. It is the same system as running the buffer. So we create this kind of a stack where we have sponges, where we have a gel and we have membrane adjacent to it. So in this kind of a thing, we put it inside again the electrodes and then we run. So the same system by which the proteins run within a gel, it runs horizontally from the gel onto the membrane as well. So this electroblotting is done, which is very faster, and therefore we transfer the proteins on the gel onto the membrane. Now two types of membranes are basically used. One is the typical nitrocellulose membrane and another is a PVDF, which is a polyvinyl membrane as well. So these membranes have different advantages and disadvantages that are used. Most commonly in labs, nitrocellulose membrane is enough, but it has certain disadvantages associated with it. So PVDF can also be used. But basically the amount of proteins it takes up is varying, and the different conditions that you need to provide are varying. But there are a lot of advantages for which these two different types of membranes can be used. So this is again the transporting buffer system. So from the buffer system, the proteins will start moving horizontally when you applied the electric supply, and therefore from the gel it goes on to become on the nylon membrane or the nitrocellulose membrane or PVDF membrane, and the gel becomes empty. So this is the electroblotting technique. Then comes the blocking, washing and antibody probing. So the blocking is, again we talked about it in southern blot hybridization, that whatever region where the bands are not present, you cover it with, for example, salmon sperm DNA. Similarly, in here, whatever region of the membrane which is not covered by the protein bands, you have to cover with some protein, some neutral or inert protein which is there. So you use blocking proteins such as BSA, which is a bovine serum albumin, or non-fat milk, or gelatin serum. You can use that, and in this inset you can see what happens, that this is going to occupy all the parts of the membrane which do not contain any protein. So it will leave just the protein bands which are exposed, otherwise everything is covered by this. So this is called as a blocking agent, so that when you add your probe, the probe binds to, or only the protein bands which are on the membrane are exposed to the probe and not the rest of the membrane system. So you know this is how it is basically done in the lab. This is snapshots of how it is done. So you take the membrane, you add the blocking agent, and you shake it on a simple shaker, so that it takes a little while, it just keeps shaking, rocking, and then the entire system is covered with that. Then comes the part of washing. Now washing steps are again very necessary to remove any unbound reagents and reduce the background, thereby increasing the signal to noise ratio. The buffered saline and phosphate buffer saline are the most commonly used wash buffers. So we have to give multiple washing steps, so that any excess amount of BSA, serum, etc., it just get removed and only the necessary amount remains. So the washing steps become very important. Occasionally, the wash buffer formulations consist of a detergent such as Tween 20 to aid in the removal of non-specifically bound material. So we actually do not want any more non-specific bound material, so we do these washings. And then after that comes the step of the probing. Now there the probe was again a DNA molecule in southern blot hybridizations, or an RNA molecule that we used to probe with. But for proteins, the probe that is used are antibodies. So the protein which is on the membrane act as the antigens. So it can detect the entire antigen or certain epitopes of the antigen. So and these antibodies are added, and these antibodies are raised against the target protein which are the antigens, and therefore it will specifically go and bind to our protein of interest, because we are going to use the antibody which is against the specific protein. Now if we are using common proteins, if we are using to identify certain general proteins, antibody may be commercially available for them, and therefore you can just buy that antibody. This is called as a primary antibody, the first antibody which we add, which goes and bind to the antigen or the epitope or a part of the antigen which we want to identify. But if you are talking about, or you are basically studying a specific protein, the antibody may not be commercially available, but you can inject that protein into mice, and then you can raise these antibody and isolate those antibodies and then use it for Western blotting. So obviously it requires, there's a protocol for antibody generation in mice. So you give injections of antigens, you give a booster dose after 45 days, then at the end you euthanize the mice and you isolate the antibody from the serum, and then this antibody you ascertain what dilution to use, and then also you can use it to detect a particular or specific protein. For example, you want to localize a protein, your protein of interest, whether it is present in the cell or cell lysate, the cell is secreting it or not secreting it, then you can raise the antibody in mice for your particular antigen. But if you are generally studying a common protein, then the antibody may be commercially available as well. So antibody is diluted based upon the concentration of 1 mg per ml. So 1 is to 100, 1 is to 10,000, 1 is to 1000. You have to standardize this, and you basically do it in the buffer system, and then you add the antibody. So this is the same thing that they are doing. So we are adding the antibody and we keep rocking it so that the primary antibody goes and bind to the antigen system. So this happens really quickly, but again, we give some time so that the antibody binding takes up. The most two important standards while choosing an antibodies are whether it can recognize the protein and whether it can cause the cross reaction. So these are the two considerations that we need to keep in mind. One thing is that it should be specific to the denatured proteins or your gene of interest. It should be able to recognize that. And second is that there should be less of a cross connection. It should not identify another similar kind of a protein which can be present. So these are the two major considerations. Any kind of antibody, polyclonal, monoclonal, or a mixed type of monoclonal antibody can be utilized. Obviously, some has certain advantages but certain other disadvantages which are there. So the polyclonal antibody has certain advantages, but it also have certain, so it depends upon purpose, that what do we want to do. But obviously polyclonal antibody or the mixed monoclonal antibody is the best to be utilized for the identification of your target protein and to avoid as much as cross reaction that can occur. So these are the antibodies. Now when we talk of detection methods, now we talk of two different kinds of antibody system that we use. One is a primary antibody system. So if your primary antibody is tagged or labeled with any kind of, because it is acting as a probe, so antibody is tagged with any detection system, then it is termed as a direct method. However, generally we employ an indirect detection method. So in the indirect detection method, the primary antibody is recognized by a secondary antibody which is then tagged or labeled by a particular detection system. So we'll talk about different detection system as we talked about in the southern blot hybridization. There can be bioluminescence, chemical luminescence, there can be radioactivity, all these different kind of tagged system. So when we use secondary antibody, it is termed as the indirect method, and when the primary antibody only is tagged, it is called as the direct method of detection. Again, they have advantages and disadvantages. The direct method, where we only use primary antibody, you require only one antibody, which is the primary antibody, and you do not require any other antibody. And therefore, the cross reactivity is less because the primary antibody will be really specific target to the proteins, your protein, target protein which is there. However, it has certain disadvantages because you have to label the primary antibody, and this label may interfere with the target binding because the primary antibody is only targeting, so it may affect its targeting capacity. Therefore, that can be a disadvantage to it. Then you have to produce also. It can produce some kind of a high background as well because of the non-specificity for a target tissue. The conjugated primary antibodies may be costly. If you prepare, conjugated means addition of a label, conjugated to a particular label. The production of this is costly, and the selection of conjugated primary antibodies may be limited. So because commercially they may not be available, if you prepare your own primary antibodies, then to conjugate it becomes another task. So direct method, sometimes it is advantageous, but more commonly the indirect method is used. So in direct method use, there is a signal amplification, as you saw here. One primary antibody was identified by multiple secondary antibodies. So in here, if there was only one signal, in here for the same binding you are having two signals. So just to very simply explain, so there is a signal amplification. You will got a robust signal with the secondary antibody system. So it is also one of the advantages for that. Then again, there can be a number of secondary antibodies that can be raised. for different primary antibodies it can detect. So the secondary antibody, because it is actually identifying this constant region. If you go through the immunology lectures, then there is a constant region which is there. Because this is identifying the constant region, it has nothing to do with the target protein. Therefore, you can have the same secondary antibody can work for many different primary antibodies, because you are tagging the constant region and not the target specific epitopes or the region which is there. So it has got many different advantages which are there. The secondary antibody, obviously it will reduce the cost as well, because the primary antibody and the secondary antibody is used, which you can use from a common pool. The important aspect is that the specificity need to be ascertained. So wherever the primary antibody is raised in a species, the secondary antibody raised in that species will work the best. There are certain disadvantages, as it also produces non-specific binding, because you are having an additional antibody which is used, and there can be some additional costs which are required, or actually the steps. The cost is not much more, because a common pool of secondary antibodies can be used to detect primary antibodies as compared to conjugating a primary antibody, which is much costlier, but additional steps are required, because you add primary antibody, do washes, then add secondary antibody, do washes, and then go in for the detection. But more commonly the indirect method is used. Coming to the detection methods, there can be many different types of detection based upon what kind of labeling for the probe one has done. So this is, for example, your protein of interest. This is the primary antibody. This is the secondary antibody, and this is the kind of tag it is covering. So there can be a bioluminescence which is there. So it will produce certain kind of a light in accordance with the product that it is producing. So with the production of the product, there can be there is this is termed as the bioluminescence. There can be a chemical luminescence as well, that the substrate is converted into a product which is giving some rise to some kind of a light, which can be detected at a particular wavelength or by certain colors which can be there. So there is a bioluminescence method, there is a chemical luminescence method. There is autoradiography. We discussed about the autoradiography in a southern blot hybridization method as well. So what happens in autoradiography? That the secondary antibodies are tagged with radioactive molecules. Now these radioactive molecules are going to produce the radioactivity, and this radioactivity can then be detected. We can develop an X-ray film. When we expose the membrane to the X-ray film, this radioactivity reduces the AgNO3, the silver nitrate, which is present in the developing solution, and therefore the reduction of the silver creates a black band. So the autoradiography can be used to detect the samples. Similarly, the immunogold method, another important method, where the gold particles are attached to the secondary antibody. Now these gold particles can again be detected at a particular wavelength. Although they give a particular color, so they give pinkish color. However, that color is not permanent, it is not stable. Therefore, another method is used, that it is developed in accordance with the X-ray, just like autoradiography, because this immunogold particles can also cause the reduction of silver, and that reduction of silver gives that brown black kind of a pigment which is there. So this is the immunogold detection method which is also employed. So various detection methods depending upon the tag or label that the secondary antibody is carrying. So what do? We isolate, just reviewing the entire procedure, we isolated the protein. We isolated the protein from different cells, tissues under different conditions, etc. That total protein lysate, we prepare the sample, we add beta-mercaptethol, we heat it, the sample, and we add SDS so that they are reduced to single polypeptides as well as masked by a negative charge all through, and then we fractionate it on SDS page, which is polyacrylamide gel electrophoresis. After electrophoresis, we electroblot the protein bonds onto the nylon membrane, and then we do different washes, we add the antibody probe, and then we detect the antibody probe, for example in this case an indirect method where we use a secondary antibody to tag that primary antibody. So that is basically the detection method, and this is the Western blot analysis of a protein molecule. So as you can see in the SDS page, you have got a series of proteins which are there. Now this series of proteins, so this is a HeLa cell lysate. So HeLa cell line, there is a kind of a cell line which is used, and you have prepared the total cell lysate, and you have basically different concentrations. So amount of total lysate is varying out here. So you add the total lysate. Now this is a resolving gel, or there was a stacking gel on the top, and then there is a resolving gel in between, underneath that. All the polypeptides which was contained in this HeLa cell, this is a total cell lysate, have fractionated on this STS page gel. You can see the different kind of bands. You can also see the different intensity. This is stained with Kumasi blue. So this is a typical gel, how a protein gel looks like. Now when you probe it with this particular antibody, the antibody against CDK7, so this antibody CDK7 and detect by chemiluminescence. So there is a chemical luminescence, a color change, that is production, which you can observe here. So you add the antibody against CDK7, the primary antibody you added onto it. So you first created this gel, you electroblotted it on the nylon membrane, and the nylon membrane is basically you treat with different washes and different things, and you add the antibody, you again do the washes, and you detect the chemiluminescence detection. So you can very well see a single band out here of different intensity. Different intensity because the concentration of the protein was reducing from the left to right. So it is reducing from here to here. So you can see a darker band and a smaller and a lighter band. So lighter band is basically the concentration is less. This is a typical Western blot that is generated. So in this particular cell line, the CDK7 is detected. So one thing, as I told you, was the detection. So if there was no band, and we obviously use a positive control so that to ascertain where this CDK7 comes in. So if we have used a positive control, and if we have used the protein lysate from a cell where there appears no band, so if this band is not there, so the CDK7 is not produced in that cell line. That is basically the detection of a protein in a particular cell or a tissue or whatever, or different conditions. For example, we used a normal cell and we use a diseased cell. So any kind of a disease, a muscle wasting disease, or any kind of a disease that we are taking. So we take a muscle tissue. So we take a muscle tissue, a normal muscle tissue, and a tissue from a person which is suffering from, say, muscular dystrophy. So we take that protein. So when we use the proteins, we prepare a total lysate, we add a particular protein that we want to identify. For example, if we want to identify a particular protein, say TNF alpha, it is a cytokine, or tweak, it is a TNF like weak inducer of apoptosis. So if we add those antibodies against these compounds and we check what happens in the normal cell line, what happens in the diseased cell line, then we see either there will be an increase in expression. So in the normal cell line there was no expression of tweak, but in the diseased cell line there you can see the band for tweak, and obviously you have a positive control, so you can identify that okay, this is the protein that you have used to create the primary antibody, for example, or it is available, so you can use the purified available tweak protein as well. So what you get is, so you can compare the different disease, so that is basically to know the different, so you can know the size, you can talk about the expression or the production of different proteins, whether it is occurring or not, and of course for the detection, whether it is present or not present. So typical Western blot occurs just like this, and you can know more about the biomolecule, which is the proteins that are present or not present. So we have discussed upon the blotting techniques. The blotting technique is basically dealing with the transfer of the biomolecules onto the inert supports, so that we can detect a particular fragment of that biomolecule or a particular part of that biomolecule which is our interest, what we want to manipulate, whether it is DNA, whether it is RNA, or whether it is protein. We want to carry out certain downstream applications. I will talk about more of the applications of these techniques in our later sessions. But if we want to manipulate them further, or we want to use into a biotechnological techniques downstream, we need to identify, we need to detect, we need to know the location or the presence of these biomolecules. And for that, what we do is we identify them, we detect them by using the technique of blotting and hybridization. So by blotting, first of all, we identify the biomolecules, we run it on gels. So isolation of these biomolecules is different. So we isolate the biomolecules, we run it on different gels. For example, in case of protein, we run it on an SDS page gel. For DNA and RNA, we run it on agarose gels. We run it on different gels. We blot it onto the membrane. So again, this blotting can be a simple capillary action or it can be electroblotting method. And when we get it here, we use the probe, which is the hybridization process. For the hybridization, we can use DNA. We can use the part of the whatever we are interested in, we need to specifically target that. So the complementarity base pairing works in case of DNA and RNA. So you make the complementary probe, which is either you synthesize it or you isolate it from some other species or some other organism, and then you single strand it and you add it to the membrane. It is going to go and bind to the membrane which is present there. And in case of proteins, you use antibodies. So for this antibody, again it is a specific target protein, it detects that protein. So that constitutes the hybridization part. So for the hybridization, you detect these hybridization. So wherever these DNA probes or the RNA probes or the antibodies, they are binding, you detect it with some system. So some system where the labeling of the probe, whether it is the DNA fragment or whether the antibody, is by autoradiography, the radioactive probes, or by non-radioactive detection methods where there are chemical adjuvants that are basically conjugated to the probes, or there can be any different kinds of methods that are utilized that we have discussed in our last two sessions. So southern blotting, northern blotting and western blotting, they are very important for usage in the different kind of biotechnology techniques. We talk about again another detection method, which is the PCR, the polymerase chain reaction, which is used for detection but more so for the amplification of the gene of interest. So amplification means making the copies of a particular gene of interest. So if we want to amplify, obviously amplification also means that it will detect. If it is present, then only it will amplify. So for detection, this blotting and hybridization was used, but for detection, again the PCR amplification can be used. So we discuss about the PCR amplification in our next session. Thank you.